This is the cell we're going to be working with today. So this is the cell membrane, the nuclear envelope, the DNA, and the centrioles. So interphase is basically just an extended period between cells. So cells are actually in interphase the longest. So the first part of interphase is actually gap 1, which is also known as G1, and this is when the cell is going to grow. So as you can see, the cell membrane got bigger. And then the proteins that are necessary for the division of the cell are actually going to be made in here as well. So then the cell goes through a checkpoint. So the checkpoint is basically just making sure that the cell is large enough, has sufficient nutrients, and then it has enough growth factors present. So if the cell either doesn't make it to the checkpoint, it can also go through G0, which is a stable stage. So it's a non-dividing phase, which is basically just like a waiting period to go to the checkpoint in order to get into the synthesis phase. So the waiting period is due to regulatory signals. So the next phase would be the synthesis phase, also known as S phase, and this is when the DNA is going to be in its uncondensed form, as you can see now, and then it's actually going to be duplicated, so there's going to be 2 sister chromatids. These are the 2 chromosomes, and then when they are duplicated, it'll be attaching by the centromere, which is right here, and then these are going to be the 2 sister chromatids. So still in its uncondensed form, it's going to enter the gap 2 phase, which is G2. Basically, there's going to be more biochemical reactions, and it's going to be preparing for division. So it's basically to check and see if they have if the cell has all its components, and that the DNA is duplicated. So when discussing meiosis, it actually goes through 2 different stages. So meiosis 1 occurs after interphase, which is the same cell cycle of interphase that happens in mitosis. So this is what the cell is going to look like with the cellular growth, as in G1, the DNA that's duplicated that happens in S phase, and then just the preparedness for eventually separation that happens in G2. This is what the cell is going to look like. It still has the nuclear envelope, the centromeres, then the DNA that's been duplicated. This is the start of meiosis 1, so it's going to start out in prophase 1, which is going to have the homologous chromosomes pair up, and it's going to form these tetrads. So this would be a tetrad, and this would be a tetrad. So then the chromosomes are basically going to begin to condense, and then there's a synapsis, which is the pairing of the homologs, which is what this is right here. Another thing that happens in prophase 1 is the spindle fibers are going to start to form, and then crossing over can actually take place. So what crossing over is basically the trading of pieces of chromosomes between a non-sister chromatid. So that means these 2, since they are not a part of the same chromosomes and they're non-sister chromatids, they can basically trade pieces of the DNA. So the other thing that happens in prophase 1 is when the nuclear envelope is actually going to break down. So this is it breaking down. So there actually next would be a pro-metaphase, but it's not really talked about that much. So we're going to move into metaphase 1. So in metaphase 1, this is when the homologous pairs of chromosomes are going to line up on the metaphase plate. So next would be anaphase 1, which is when the homologous chromosomes are going to separate and move towards the opposite poles. So they're doing this with the spindle fibers that are drawing them back from where the metaphase plate was and drawing 2 chromatids of each homologous chromosome over to each pole. Next in meiosis 1 would be telophase 1, which is when the chromosomes arrive at the spindle poles and the cytoplasm starts to divide. So this would be representing the division of the cytoplasm, and then the nuclear envelope that's not actually fully there in telophase yet, but it is actually starting to form around each gonna be new cell. So after telophase 1, the next step is going to be cytokinesis in the meiosis 1 stage, and basically what's going to happen is the chromosomes are not going to be condensed anymore, and the cytoplasm is going to divide into 2 daughter cells. And then, as you notice, the parent cell had 4 chromosomes, and now each daughter cell is only going to have 2 chromosomes. So the next stage is going to be prophase 2, starting meiosis 2, basically. So in prophase 2, the chromosomes are going to recondense, and the spindle fibers are going to start to develop. This is also where the nuclear envelope is going to break down in prophase 1. Next is going to be metaphase 2, which is when the individual chromosomes are going to line up on the equatorial plate. So it's right down the center like this, and these spindle fibers are going to attach to them. So the next part of meiosis 2 is going to be anaphase 2, and this is when the sister chromatids are going to separate and move towards opposite poles. So as you can see, these two sister chromatids separated, and they move towards this pole and this pole, along with the rest of both cells. So next would be telophase 2, which is when the chromosomes are going to arrive at the spindle poles, which we've already seen, and then they're going to start to have the cytoplasm divide, which is what this is representing, along with like the nuclear envelope starting to reappear. So lastly, these cells are going to go through cytokinesis, which is going to be them dividing completely into their own cell. So there's going to be the end product of 4 haploid daughter cells, and they are not going to be identical.